Alright, in this video I'm going to go through the biosynthesis of thiamine, otherwise known as vitamin B1. So, thiamine, and I've seen it spelled with and without the E, but you can also just call it vitamin B1. And so this is somewhat of a complicated biosynthesis because it doesn't just follow a nice linear pathway. And so for this one, there's this pathway right here to generate this HMP pyrophosphate, the 4-amino 2-methyl 5-hydroxymethyl pyrimidine phosphate. And the name can be a little confusing for that because it talks about this hydroxymethyl, and it doesn't look like there's a hydroxymethyl, but they are talking about it as if there is no pyrophosphate on it. So this here is a hydroxymethyl, and so it's hydroxymethyl, and then this is a pyrimidine, and then phosphate, or in this case pyrophosphate. So it's 4-amino, which is this, 2-methyl, which is this, then 5-hydroxymethyl, which was this, and then pyrimidine phosphate or pyrophosphate. So the first step here, this THIC just generates the phosphate version. THID just adds another phosphate on to make it pyrophosphate. And so I'm going to talk about this step here, but I'm not going to go through this step since it's just the addition of a phosphate, which is a pretty common mechanism. And then there's this other branch here, which is further split into its own 3 branches. So there's this one up here, which takes either L-tyrosine or glycine and generates this dehydroglycine. And the L-tyrosine is done with THIH, or the glycine with THIO. So this isn't, you don't need both of them at the same time. This is or one or the other here. So what it's trying to generate is this dehydroglycine, which is pretty easy to see how it comes from glycine. It's just so this has 2 hydrogens on it. So just be kind of the extraction of a hydrogen and generating that double bond. For the tyrosine, you actually have to just remove the entire phenol group and just end up with this to generate the dehydroglycine. Another one of the branches is this DXP, generating this DXP from pyruvate and glyceraldehyde 3-phosphate, both of which come from sugar metabolism, which I will get into more in future videos when I go through glycolysis and so forth. But these are products from sugar metabolism, and then through this DXP synthase enzyme, which puts these 2 together and removes a carbon dioxide, we end up with DXP. And this is something that does not happen in humans. So this is one of the reasons that that humans don't produce thiamine. And then this third step, which is a very complicated one all in itself, because it requires this ISCS, this THIF, this THIS, and this THII, which then when that comes together with these other ones, with this THIG, there's this sort of handing off of these intermediates throughout these. So these enzymes all sort of come together into these complexes while handing off these different intermediates to each other, and sometimes even combining the intermediates in the enzyme complex. And so I'll go through that and hopefully make that a little bit more clear. But then taking the S carrier, which is this THIS, which can also be bound to the THIF. So like I said, it's a kind of a strange mixture of handing things off and so forth, which we'll go through in a little while. But then this THIG then sort of brings all three of these things together, this one, and this one, and generates this thiazole ring. And then there's an extraction of this hydrogen and generating and sort of moving these double bonds around with this 10I here, this TENI, to generate our thiazole phosphate. And then we have this PHIE, which brings our HMP pyrophosphate and our thiazole phosphate together into thiamine monophosphate. Then this THIL phosphorylates that into thiamine diphosphate. All right, so I'll start by going through the THIC mechanism here. And this one uses ferredoxin and s-adenosylmethionine. So the s-adenosylmethionine, which is so named because it has this this adenosine here. So this is an adenosine moiety here, and then a methionine moiety here. And so this S is positively charged, and so this is often used for methylation. This methyl group right here, because it's on this positively charged sulfur here, can be added to other things, including DNA. So if you've ever heard of DNA methylation, then this is the methyl group that gets added to that. But in this case, we are not getting this methyl group. We are actually using this ferredoxin, this iron, what's sometimes called cubane. So cubane here because it makes this cube, this iron sulfur cubane. And so using this, it's actually going to generate, so it's going to detach the methionine from it, but then also generate this radical. So this 5' D-adenosine radical. And so this goes through, uses this mechanism here. So the methionine moiety actually coordinates with one of the irons on this ferredoxin. And then this iron here transfers an electron here so a single electron onto this sulfur here. And so a single electron can then get moved over. So I think this arrow is pointing at this carbon here, but it's actually moving to that carbon there. And so we end up with a radical on that carbon that would be this carbon here, while the methionine stays coordinated with this ferredoxin. And I actually have the structure here for that. So we can see down here, this is our ferredoxin, this yellow-orange cube thing. And we can see it's held in here by these cysteines, which is what this showed. It has these cysteines on it. And we can move over here. So this is, well, this is kind of two things overlaid with each other, but you can see over on this side where there's this salmon colored thing here. This is our methionine. And then over here with these ring structures are adenosine. And so you can see that in both cases, this is brought close to that ferredoxin here. So I will get rid of this homocysteine here, this S-adenosylmethionine homocysteine. So we can see our methionine here. Interestingly, this shows it actually coordinating here with this zinc. Yeah, it's a zinc ion here. rather than coordinating with the ferredoxin that this proposes is what's actually happening. But in this structure, it actually looks like it is coordinating with the zinc, whereas the sulfur here is near the ferredoxin. So if I bring that S-adenosylmethionine homocysteine back, and let's remove that methionine here. So this is our S-adenosylmethionine homocysteine, which is similar to the S-adenosylmethionine, except there isn't that methyl group coming off of this sulfur here. But we can see how that is fitting into this active site. And once again, it seems to be coordinating with this zinc rather than the ferredoxin as proposed in that mechanism. Another interesting thing about this protein is it's a homodimer, so it's two of the same enzyme brought together, but this one that's in this purplish color is actually the one binding the ferredoxin here that is brought into the active site of the green one. And so there's this interesting topology here where one of the monomers in this dimer is holding onto the ferredoxin that's used in the other monomer here. So that is just the first part of this mechanism, is generating this 5' D-adenosine radical, because then that is going to be used in this next step, which takes this AIR, which is 5-aminoimidazole ribotide, and uses that on this 5-member ring here to sort of turn it into a 6-member ring right here, which is our HMP pyrophosphate, or HMP phosphate rather. Pyrophosphate not till the next enzymatic step, but so this actually comes from purine biosynthesis. Purines being bases in DNA. So purine biosynthesis, one of the intermediates in that is this 5' phosphoribosylformal formal glycinamide, and then that this is closed into a ring which forms this 5-member ring here. Then the THIC, using the 5' D-adenosine radical, will turn it into this 6-member ring here. And I had a hard time finding a good mechanism for this, and the one that I found that I then just sort of copied and added my own arrows into was one that didn't actually have arrows. It just sort of showed intermediates. So I drew up what they had in ChemSketch and then added my own arrows based on what I believed they were, the paper I was using thought the mechanism was. So I'll go through that here. So this is our 5' adenosine radical. This is our 5-aminoimidazole ribotide. So that is this species up here. And so I'm showing this with the radical already generated here. And so what I thought the mechanism was, based on just the images of the intermediates, was that this radical here went and snagged this hydrogen off of here, and so one of the electrons from this bond goes to generating the bond, turning this into a CH3, and then one of the electrons ends up on this carbon here. So over here we've generated our CH3 by snagging that hydrogen off of there, and now we have a radical here. And then this next step, they just kind of showed this structure here, and then they just kind of went to the next one where this OH, where this oxygen was no longer bound to this carbon. And so I thought maybe this could be a concerted mechanism, which I drew over here. And so the electrons from this double bond going and attacking this carbon here, so that the electrons from this bond here go towards making a bond there. And then these right here, these electrons in this bond here move over to this bond here. And then the electrons on this nitrogen here move to make the double bond there. This double bond moves over here. And so that's how I thought maybe you could end up at this step, sort of just skipping over this step and doing it as a concerted mechanism. So you end up with this five-member ring detached. We have our double bond on the oxygen here, which was generated from that step. And so, yeah, I thought maybe that could be concerted, or maybe there is some acid binding to this oxygen here to sort of detach it from here. But then there's just a lot more charge bookkeeping one would have to do. And so I'm not exactly sure if it would be this concerted mechanism or not. But anyway, we end up with these intermediates, and so we have this double bond move here, this move here. Our electrons are on this carbon, making it a carbo anion, and then maybe this comes down and attacks this carbon here. It seems kind of a strange one since there's no charge on this, but this is the next step that was shown in the mechanism I found, where this five-member ring here is sort of moved over on the so this nitrogen to carbon bond is broken, and instead this carbon is then bound to this carbon, like we have shown here. So, and then this next step, I have this radical here going down there, an electron from that bond going there, and one going to that, so that we end up with this radical, and this is no longer a radical. But then we could have the hydrogen from here. So then this radical is moving down there. This is going up there while this comes here. So we end up with a radical sort of move from this carbon here over to this carbon, maybe using a residue in the enzyme. And then the mechanism I saw sort of just moved on to this this three carbon cyclic structure here. So I proposed this mechanism here where one of the electrons moves onto this nitrogen, and then one of the electrons moves and bonds to this radical here, so that we end up with our three carbon, our cyclic three carbon ring here. And then the electrons in the bond on this three carbon cycle here move over to so that this forms another double bond here along with this radical on the nitrogen, where one of the electrons moves over to form this radical here. And then we move on over here where this radical then moves down here. One of the electrons from this bond goes along with it to form the double bond, while this one goes over here, detaching this moiety from the structure here. Then we have another radical on here, which goes to form a double bond with this oxygen, where the radical moves down to this carbon here, and there's a double bond forming, or or not a double bond, but this is actually detaching. This whole hydroxyl group is detaching. So we end up with a water coming off, and now we have this structure here, and then the radical on here can actually bond with this carbon here, so that we end up with this structure. Then our water that came off can then come over and attack this aldehyde carbon here, and so we end up with this dihydroxy moiety here. But then this comes down and forms a double bond on between this carbon and oxygen here, while this bond here detaches, and the electrons for it sort of snag that hydrogen off of there, so that we end up with this structure here. And then we have an electron coming probably from that ferredoxin here, coming down to generate a double bond along with this radical right here, and that's how we end up with our HMP phosphate. Yeah, so that is the mechanism that I proposed for this. So I put this down here once again just to remind us why it's saying hydroxymethyl here when there doesn't look like there's a hydroxymethyl on here. And in fact, in ChemSketch, I drew this incorrectly. This phosphate here should actually be bound to an oxygen, which is bound to this carbon. So that was a mistake on my part, going quite a ways back here. Yeah, that was my bad with the ChemSketch. So it should actually be a phosphate. So it should be there should be an oxygen bound to this phosphorus right here, and then that oxygen is actually what's bound to this carbon right here. So it would be kind of like on this, except instead of this H, we would have our phosphate here. So yeah, if you caught my mistake there, then yeah, that good on you, because I screwed that up on here on ChemSketch. But it didn't really change the overall mechanism here by that much, because this phosphate wasn't really active in our mechanism here. So the mechanism should still be the same. It's just that that phosphate, as I said, should have another oxygen bound to it. But anyway, that is our THIC mechanism. So to go back here and sort of get our bearings a little bit, that generates this species here. Then a THID would come and add another phosphate onto it, turning it into HMP pyrophosphate. So now let's go ahead and look at this THIH, THIO enzymes mechanisms over here. Yeah, by the way, this is the eukaryotic pathway. So I'm pretty much going through just the prokaryotic pathway. It's a little bit better characterized in the literature, and it's essentially the same, where in the eukaryotic we are generating our thiazole phosphate and our HMP pyrophosphate, and then bringing it together into the thiamine monophosphate. And in fact, this enzyme here, this THI6, says it has the THIE domain, which we won't see in a little while when I look at that structure. And it's the THIE that's bringing it together here. So it's the same mechanism in this enzyme. But let's get back to this THIH here, which is to generate our dehydroglycine, which is this species here. And so the structure I have actually shows a tryptophan in it, but um, the reaction mechanism would be the same essentially. And so we're taking tyrosine here and using another s-adenosylmethionine radical. Just like we did with the THIC, we are generating a radical here. And so the proposed mechanism at first thought that the radical was being generated here on the phenolic oxygen, and then sort of moving its way through this aromatic ring to come down and detach at this bond here to generate our dehydroglycine. But a later paper that I found using what's called electron paramagnetic resonance to look at the placement of radicals in an intermediate. So that's what this EPR is the electron paramagnetic resonance. Actually found that the radical is first being generated on the nitrogen here rather than on the oxygen. And so using this radical mechanism here, the radical is moving down here. One is moving down from this bond that's being broken, and then the other one is moving to this carbon right here, so that we end up with this this 4OB, which is 4-oxidobenzyl. It's a 4-oxidobenzyl radical. So we see our radical ended up on that carbon there, and we end up with our dehydroglycine. And so the structure I got, which wasn't for THIH, was actually for something called HYDG, and so it does this same mechanism to generate this dehydroglycine, but then it does another mechanism which actually generates this carbon monoxide and this cyanide. So I'm pretty much going to be paying attention to this first part here. Uh, so that is this enzyme right here. And so we can see we have another of this ferredoxin, this iron sulfur cubane here. And we can see we have a methionine. So this one we can actually see that methionine is coordinating with the iron on here. So the carboxylic acid and the nitrogen on that methionine are actually coordinating to our ferredoxin, which, as we saw in the THIC, didn't seem to be the case, where it looked like our methionine is actually coordinating over here with this zinc ion. But in the this THIH analogous enzyme here, or homologous, maybe you could say, it is actually coordinating with our ferredoxin. And once again, we can see these 3 cysteines holding the ferredoxin in there. In yellow down here, we see that we have tryptophan rather than tyrosine. So this could happen to both tryptophan or tyrosine. But you can imagine that there could be a tyrosine in here, and it makes sense actually if you think about the this other mechanism where we're actually forming the radical on the nitrogen rather than the phenolic oxygen there, because this tryptophan doesn't have a phenolic oxygen. So if we wanted to detach a dehydroglycine from it, it would actually need to put that radical on the nitrogen, since this does not have a phenolic oxygen. In this structure too, we actually have this second ferredoxin over here. And this ferredoxin actually has another sulfur and iron bound to it. And this actually has the dehydroglycine here on it, with this extra irons being held in with this histidine. And this is where that second step would come from, here in the HYDG, but that is not a step we're interested in here because we just wanted to form this dehydroglycine here, and that happens over in this part of the active site. So the THIO, which is just turning the glycine into dehydroglycine, instead of using that ferredoxin, is actually using this FAD, this flavin adenine dinucleotide, which is what I have in cyan here. And this in yellow is actually an acetylglycine, and that they use in this structure so that it can actually go into the active site without reacting, so that they can actually capture the structure of it in the active site here. But you can see that this is the carboxylic acid here, and the nitrogen here, and the alpha carbon, then it just has this acetyl group on it. So we can see how this glycine would come close to this FAD, this flavin adenine dinucleotide, and these nitrogens and so forth will be exchanging electrons with this in order to sort of remove one of the hydrogens on here so there would be two hydrogens right here. One of them will be removed and one from the nitrogen will be removed. The electron's going to form that double bond to generate dehydroglycine. So we're removing hydrogens. That's why it's called dehydroglycine. And so that is the two ways we can end up with our dehydroglycine is through this this tyrosine or this glycine mechanism here. All right, to go back and get our bearings again, we've now generated our dehydroglycine and our HMP pyrophosphate. So we still need our DXP and our sulfur here. So we will first go through this DXP, which stands for 1-deoxy-D-xylulose 5-phosphate synthase. And so this, interestingly enough, actually uses a thiamine as a cofactor. So we have our thiamine right here as our cofactor. So to generate thiamine, we need thiamine. Well, not humans, not we. Bacteria use this thiamine here as a cofactor. And so thiamine has this acidic carbon right here, so hydrogen can be extracted off, and we have this negatively charged, this carboanion here on the thiamine, and that will attack this carbonyl carbon here on the pyruvate here. So this species right here is pyruvate. So we end up with this intermediate here, this covalent intermediate here. So this attacks this, the electrons move up to this oxygen here, so we end up with a hydroxyl there. And so we have this covalent intermediate, but then the electrons on this carboxyl group here go down, and these electrons on this bond break off and go down to form a double bond here. The electrons in this double bond move onto this nitrogen here. So we end up with this intermediate after this decarboxylation step. So we are losing this CO2. And so the structure I have for this, so this is the THIO, this is the DXP right here. So we can see here in yellow, well, in salmon is the pre-decarboxylation intermediate, and the yellow is the post-decarboxylation. So the salmon is actually an analog. So instead of going to an oxygen right here, it goes to this phosphate, which then has a methyl on it. but this would have the sort of same structure as if it had a carboxyl group on here. And so this is being removed. So I will remove the post here so you can get a better look at that. So this is that, so this right here is actually our thiamine, and then it's bound down here to this, and then this is our carboxyl analog right here. So this fits into the active site like that. The pyrophosphate on our thiamine is coordinating with this magnesium here, as is pretty common. The carboxyl group is held in place here by these two histidines. And then if we remove the pre-decarboxylation and add in our post-decarboxylation, we can see we have now bound to our thiamine this group right here, which is this group right here. So this is that methyl group, this is that hydroxyl group. We can see that here, that methyl group, that hydroxyl group bound to our thiamine right there. And we can see that this histidine is sort of holding on to this carbonyl oxygen right there. It looks like with this phenylalanine here, we might have some pi stacking going on with the pyrimidine ring on the thiamine right there, and maybe even some more pi stacking along with this tyrosine right there. So that is sort of this step here, the pre-decarboxylation, the post-decarboxylation. But then this glyceraldehyde 3-phosphate, so it's glyceraldehyde, which is all this here, then has a phosphate on the third carbon. So glyceraldehyde 3-phosphate comes in. This negative charge here on our intermediate then attacks this aldehyde carbon here. So we end up with another covalent intermediate, but then the electrons on this oxygen come down to reform a double bond on here, which causes this bond to break. The electrons going back onto our thiamine to regenerate this carboanion species. Now our DXP is generated there. And so we can see here that we have these right here coming from our glyceraldehyde 3-phosphate. Actually, it would also contain these ones, wouldn't it? And then this coming from our pyruvate. So that is our DXP. And the paper I saw had these kinetic parameters here. I don't know if you can see that very well on your screen, but it just shows that the Km for the pyruvate is 54 micromolar. The Km for the glyceraldehyde phosphate is 11 micromolar, and the Kcat is actually somewhat slow. It's about 45 turnovers per minute. So something like 1.25 per second. So that is our DXP. So go back here to get our bearings again. We've now generated our dehydroglycine, our HMP pyrophosphate, and our DXP. So now is the sulfur carrier. And this is a bit of a complicated few mechanisms here. So the first one here is we're sort of stealing sulfur off of cysteine and binding it to this enzyme called ISCS. And then this ISCS is involved in a bunch of other things. So the one we are interested in is this one up here. So this handing it off to this THII. But I also wanted to show here a few of the other things. So this is one of the other things that sulfur can be used for if our T8 or if our ISCS hands it off to things like TUSA or this ISCU and so forth. And for that, it's for generating things like our iron sulfur, like our ferredoxin, like this molybdenum cofactor, but also these nucleosides that have these sulfurs, these post-transcriptional modifications, putting sulfurs onto them in the mRNA. Or actually, these are in the tRNA rather. And so they put these specifically on these wobble positions here. So the anticodon in the tRNA has these what are called wobble positions in the third position here. And so those don't bind as well. And so in order to sort of differentiate between them, they do these post-transcriptional modifications. So this is actually our ISCS here bound with the TUSA or ISCU, which we see over here is our TUSA and ISCU. So these are the places they can bind on there. I put this genetic code here because I wanted to point out the wobble position is on these third positions. And so we see here that, for instance, on the glutamine, lysine, and glutamate, the wobble positions are all the same on them. And so putting these modifications on them can help differentiate between the different amino acids on there. And I put these here because this is actually the structure I found. I couldn't find one that is bound to this THII here. So I have the structure here, which is the ISCS bound to either the TUSA up here in purple or the ISCU over here in orange. But this is our ISCS, and we can actually see here in the active site. So this is actually vitamin B6. So we can actually see vitamin B6, which is this paradoxal phosphate here in the active site, and that is because in our ISCS, to sort of extract the sulfur off of a cysteine, we use in red here, this is the vitamin B6. So up here is our cysteine that we are taking this sulfur highlighted in yellow off of. So this vitamin B6 is in here as what's called a Schiff base bound to this lysine in the ISCS. Over here is sort of the general or generic depiction of a Schiff base, which is this nitrogen double bound to a carbon. In the way I always remember how the mechanism for the Schiff base goes is it's a primary amine here that binds to a carbonyl carbon or a ketone carbon to be precise. So it'll go through a mechanism like that. And the joke that I heard is, what does the primary amine say to the ketone at the party? Let's get Schiff-based. So that's kind of how I remember how Schiff bases attach to a carbonyl. So the carbonyl here that it would be attaching to would be this up here, this aldehyde is being replaced with the shift base. But anyway, the nitrogen, the amine group on our cysteine comes and attacks that the carbon double bound to that nitrogen there, so that we get this covalent intermediate here, uh, and then we end up with the electrons from this nitrogen coming down to actually form a shift base to the cysteine, where this lysine is actually detaching from the vitamin B6 here. Uh, and then over here, it just shows that our cysteine is now shift-based to this this vitamin B6 here. It shows this hydrogen is coordinating with this oxygen down here, this oxoanion. Then it shows this lysine extracting hydrogen off of just a general acid here in our active site. So that is the shift base, the formation of the shift base of the cysteine with the vitamin B6. Then the formation of the enzyme persulfide intermediate. So now after that lysine has extracted hydrogen off of this right here, it is now being used as a base to actually extract this hydrogen off of here. So generating a double bond here, this double bond moving down here, this moving over here, the electron from this double bond going onto that nitrogen there. So now we have a double bond here and a double bond here going to that nitrogen. And so the shift base kind of moved up into what used to be the cysteine here. And then this shows the electrons from this double bond deprotonating this cysteine. So this cysteine here is one that's actually part of the enzyme. It's this cysteine here that we're extracting the sulfur off of. So this double bond deprotonates that cysteine, which this cysteine then comes and bonds to this sulfur here. And the electrons move off of that sulfur. It breaks the sulfur off of what used to be the substrate cysteine, forming a double bond here. The double bond on this goes to the nitrogen. So now we have this carbon double bond carbon here. The double bond on that then extracts the hydrogen. It deprotonates that acid once again. And then over here, now that this acid has been turned into a base, so we've attached a hydrogen onto this carbon, but now it's deprotonating this carbon down here. And then this double bond is once again extracting that hydrogen off of this general acid again. But sort of the takeaway here is that we've now generated this persulfide here on our cysteine, which is the thing we wanted to do. The rest of this is sort of just bookkeeping with the rest of what used to be the cysteine. So I'm not going to go through that here in detail, but it does get released as an alanine, which so an alanine is just this COO, where our cysteine was, that should be a plus charge, had that sulfur on it, COO minus. So we've just removed that sulfur off of it, turned the rest of it into this alanine, which is released. But it was this cysteine, or this sulfur that we wanted, and we have that now attached to the ISCS enzyme. But then this THII with another cysteine here will come and actually grab that sulfur off of it, thereby sort of regenerating our active site the way it was at the beginning. and the THII now has that sulfur here. And so then the next step, so this here I guess is kind of a nice little diagram. So we took our cysteine, took that sulfur off of it, released alanine. So our ISCS here has the persulfide, but then that goes and sort of hands it off to the THII, and then this THII will go off and hand it off to the THIS plus THIF. And so we end up with the persulfide here with the THIS, THIF complex. And so what ends up happening is essentially we have the THII, which has the persulfide here. And then this goes and the THIS comes and attaches to it. And so this sort of breaks off that sulfur here onto the THIS. So this is our THIS. And then our THIF is actually bigger than the IS, so I'll draw it bigger. THIF comes and sort of displaces the THIS. Actually, that would be right here off of this, so that instead of the THII THIS complex, we sort of exchange the THII for the THIF and end up with the THIS THIF complex. And so down here, I actually have the mechanism when it's the THII and THIS complex. We have our persulfide here on the THII. This sulfur here attacks this carboxylic acid, which has this AMP bound to it. So that's why it shows an ATP here, because in an intermediate step, we actually bind AMP here and release pyrophosphate. So we have AMP here, but that will detach on this step here as we so this this right here should be our THIS. So this should be THIS. This is something else I whipped up in Chem Sketch, so that's why there's that mistake there. This should be the THIS there, right there. So then THIF comes in and sort of displaces the THII. So this sulfur binds onto that sulfur. This bond here goes to extract this proton here to deprotonate that, so that we end up with our THII sort of coming off, and now we have this THIS THIF complex here. All right, so we looked before at our ISCS here. As I said, this has the TUSA and the ISCU bound. I could not find a structure that had the THIS bound, or the THII rather, bound to it. But we can imagine it probably binds in in similar places here. The cyan and the green are just two homodimers, is just the homodimer of the ISCS. There also wasn't any substrate in the active site for the structures I found. It does have the vitamin B6 in there, but no substrate, which makes the structure a little bit less interesting to look at. I also wasn't able to find any IS or THII, but I did find this THIF THIS structure here. So the green and then this this other greenish color is just a homodimer of the THIF. Then these two cyan up here are the THIS. And so the paper I was looking at, they didn't have the sulfur substrates in, but they said that this cysteine right here on the THIF would be the one that would be bound to the sulfur. So it would be this sulfur right here on the THIF and that persulfide right there. They claim in the paper that this is the cysteine that would be involved in that. I could not find in the THIS any cysteines that were brought close to it. So they didn't determine that by the structure. They determined it through biochemical methods by mutating this out and seeing if they could pull down the THIF, THIS structure. But anyway, you can't see it in this structure here. But you can see this is an ATP down here, so that's what would be involved in putting the AMP onto this THIS here. So that is the ATP bound down there. So you can see, I thought what this looked like was almost like a face, maybe even like a Mickey Mouse head where it has ears and then sort of the head down here. And that structure is seen in related proteins. So here is the THIF and THIS. But the THIF is similar to this MOEB and this E1, this ubiquitin enzyme here. And the THIS is similar to this MOAD and the ubiquitin. So the MOEB and the MOAD are both generate another one of these sort of Mickey Mouse head structures. And this is involved in generating the sulfur on this molybdopterin here, which is involved in molybdenum binding. And so I didn't want to go too much into like molybdenum enzymes, so you can find, I don't have it in the description below because the description links are already getting quite large. So if you're interested, you can just look up this paper. You can find the whole paper online. So mechanistic aspects of molybdenum containing enzymes, if you're interested in that. So the ubiquitin and E1 are involved in this over here, this ubiquitination. So the ubiquitin binds to the E1 here. And this actually forms sort of the Mickey Mouse kind of structure too. So this is our THIF, THIS, but then I can add in the other proteins here. Where were those? Yeah, so in purple here, that's the MOAD, MOEB. So you can see how well that is overlapping on our structure here. So it's a very similar protein structure right there. And then our ubiquitin is, yeah, so it's this orange one here. And so you can see how that, the E1 down here in the larger part, and then the ubiquitin up here in the smaller part, overlap structurally very well with our THIF, THIS. And that's a little bit strange for this one, since we're not doing any sulfur transfers. It makes sense with the MOEB, MOAD, since that is also doing this sulfur transfer onto this this pyranotyrine here, putting this sulfur on. So it's a similar mechanism to our THIF, THIS up here. But we can see that evolution has also used that for this ubiquitin E1 and ubiquitin binding here. So yeah, anyway, that is the ISCS. So we looked at the ISCS, the THII, THIF, THIS. And so, as we said, we took that sulfur and sort of the TISCS extracted that sulfur, then it handed it off to THII, which then handed off to THIS, and then this bound to the THIF. And so that's sort of what this right here is also showing, is that we took the sulfur off of the cysteine. The ISCS took that sulfur off, handed it off to THII, which then the THIS bound to the THII, grabbed the sulfur, and then the THIF came and displaced the THII off of it. So now we can go up here and get our bearings a little bit again. So we talked about the dehydroglycine. We got that. We got the DXP from our pyruvate and glyceraldehyde 3-phosphate. We got our HMP pyrophosphate, and we got our S carrier. And so now actually the S carrier, let's just move on to what I have down here. So I kind of have the arrows from all these steps coming in over here. And so with our THIH or THIO, we got our dehydroglycine from the tyrosine or glycine. We got the THIS with our sulfur here. And over here we got our DXP from our DXP synthase. So these will all be brought together here using THIG. And that will be this mechanism down here. So the first thing that happens actually, so up here is just a little reminder of that THII and THIS and THIF sort of handing things off to each other. But in here, it might be a little hard to see, but our THIG comes with this this amine group on the lysine and attaches, makes a covalent intermediate with our DXP. So we're generating a shift base with the DXP here. And then so down here we have our THIS, THIF bound together. Our THII has left the building here. Then our THIG comes in and actually now forms another complex with this THIS, THIF. But then THIF actually detaches from that. And so we have our THIS, the sulfur, binding onto the DXP here. And so I actually, using ChemSketch, not a sponsor, made the mechanism here with the arrow pushing going on. So the sulfur attached to that carbon there, and so we end up with the THIS and the sulfur now covalently bound to the DXP, which is covalently bound to the THIG. So the THIG DXP sulfur THIS complex. So this kind of all bound together in this sort of topology here. But then we have this oxygen on the DXP attacking the the carbonyl carbon on the THIS here. So it's detaching this sulfur, but now we form this covalent bond with the oxygen here. But now our sulfur is attached to that DXP right there. Excuse me. And so the mechanism I thought made sense because the paper actually had something else that didn't look as intuitive to me. Uh, so I actually have sort of this SN1 um, SN1 nucleophilic uh, uh, well we'll just say that this hydrogen attaches to this oxygen, making a positively charged oxygen, so that this bond here can break, forming a double bond here. This double bond moves over to this nitrogen here, so that this species here has come off, and so has our THIS. So the THIS can now leave the building, and we end up here now with this double bond here, and this double bond here are now this one and this one. Then the sulfur comes and attacks this double bond, this carbon double bond nitrogen. So we end up with this structure here. And then this primary amine here comes and attacks this carbon right here. Electrons move up onto this nitrogen here. And now we have formed this 5-member ring. Then the electrons, the lone pair on this nitrogen here, collapse down to form a double bond here. This bond here breaks off the electrons going onto the lysine of this THIG. So now our thiazole ring can detach. And then I show here in the next step, the THIE, we will decarboxylate this to form our thiazole right there. And so, yeah, we will look at that in the next step. So we've formed this that has the carboxyl group on it now with THIE. THIE, we are going to bring the HMP-PP here, the HMP pyrophosphate, and our thiazole ring together to form this. Then this shows that it can tautomerize and then decarboxylate, or that it can happen all as a single concerted step. So I used ChemSketch to make the arrow pushing mechanism here. So we have our HMP pyrophosphate here. We have our thiazole ring here. And so we have the nitrogen on the thiazole coming and attacking this carbon here, so that this bond breaks the electrons going onto the oxygen, releasing the pyrophosphate here. We are now covalently bound together with this nitrogen onto this carbon here. Then I have this double bond sort of as a concerted mechanism going and grabbing this hydrogen here, the electrons in the oxygen collapsing down so that this CO2 will break off. This bond breaks, forming a double bond there. This double bond moves over there. Looks like I screwed up on this phosphate again. This should still have an oxygen bound here. Yeah, so it should have carbon-carbon, then an oxygen bound to that phosphate there. Once again, my screw up in ChemSketch, but it doesn't change the mechanism. There just should be another oxygen between this carbon and this phosphate here. And so, yeah, through this concerted mechanism, we now end up with our thiamine phosphate here, or thiamine monophosphate. Once again, should have that other oxygen right there. Um, and so that is our thiamine monophosphate. Then that will be phosphorylated again to generate thiamine pyrophosphate or thiamine diphosphate, however you want to say that. So that uses ATP and this THIL enzyme to just add another phosphate here onto the thiamine. Yeah, and so that is the, well, I guess we can look at the structure here. This is this is the THIG, and in purple up here is the THIS. So if you remember in our mechanism here, uh, we have our THIS and our THIG coming together. And so we can see that here, this is our THIS up here, and in green down here is our THIG. And so we can actually see the DXP here bound to that lysine in the THIG. So this is our THIG and this is our DXP, so we can see that primary, well, there's a secondary amine here because of the covalent, because they're covalently bound. But that's a lysine here, and we can see that lysine binding our DXP here. And we can see our THIS up here. We can see how that has this strand coming down into the active site. So that would likely be carrying that that sulfur, and that sulfur would then come and bind to the DXP here, on 2 carbons away from the nitrogen, and so yeah, that would be this carbon right here. So you can see how this could come in here with that that sulfur and have that sulfur bind to this carbon right here in the active site. Yeah, so that is the THIG, and then the THIE I have right here. So in salmon, I actually have the THI6 from eukaryotes, and then in the cyan is actually THIE from archaea. I do have here in green, that's the THIE from bacteria. That doesn't overlap as well with the other ones, but if we remove it, you can look in here and see. So this is our thiamine monophosphate in there, but you can see that it forms this this alpha beta barrel fold here, where we have the beta strands sort of in the center, and then these alpha strands around the outside. And if we actually add these ones, these ones in and remove that one, we can actually see that this forms that same beta sheet on the inside with the alpha helices around the outside. So they both, even though they don't overlap that well with the prokaryotic one, they do still form that same fold there. And we can see here in purple, that is our thiamine phosphate here in the active site. So that would be the product of this reaction. Excuse me. And so the THIE is kind of doing a lot of work here. It's doing both the bringing together of the thiazole ring and the HMP pyrophosphate, while also doing that decarboxylation. So the THIE is doing both of those reactions there. I'm not sure what this extra domain is on the eukaryotic enzyme. I'm not sure what that does. I didn't look into the eukaryotic as much, since I was pretty much just going through the prokaryotic pathway here. But we do know that in this figure, it does say THIE domain, and that would definitely be this part that's overlapping with the Archean in cyan there, THIE. Uh, yeah, so that is the pathway for, well, the pathways, plural, for generating thiamine, which is vitamin B1. So once again, just to quickly go over this, we needed to generate our dehydroglycine using either tyrosine or glycine, using the THIH or THIO enzymes respectively. We needed to use our sugar metabolites, pyruvate and glyceraldehyde 3-phosphate, with DXP synthase to generate DXP. And we needed to grab a sulfur off of a cysteine to have this sulfur carrier. And then that was all brought together in that complicated sort of exchange of between the ISCS, the THII, the THIF, THIS, and the THIG, along with the DXP to generate our thiazole ring. And then in the other branch of this pathway, we use our THIC on this AIR, so this 5-amino-imidazole ribotide, which is an intermediate of purine metabolism. And then using s-adenosylmethionine, we use that radical reaction with the THIC to generate our HMP phosphate, which is then phosphorylated to HMP pyrophosphate. And then our thiazole ring, our thiazole phosphate, and our HMP pyrophosphate were brought together with the THIE, and that formed our thiamine monophosphate, which is then phosphorylated to thiamine diphosphate or thiamine pyrophosphate, whichever one you prefer. Yeah, and that is, so yeah, it's, like I said, pretty much the same thing in eukaryotes. Our glycine, and we're using NAD here, and it says an S donor, so that would probably be similar to this whole pathway right here. And then this has a question mark, because like I said, the eukaryotic pathway is not as well characterized. I believe this here in this navy blue was in yeast only. I think this was yeast only part of the pathway here in the navy blue. But we are using the AIR to and THIC, which is the same as in our prokaryotic pathway, to generate HMP phosphate. Then there seems to be something that has a THID domain. So THID, which is the same as in that pathway, to phosphorylate that again. So over here we had our thiazole phosphate and our HMP pyrophosphate, that enzyme with the THIE domain, that alpha helix beta sheet barrel protein brought this together to generate the thiamine monophosphate. Interestingly, in eukaryotes, it looks like it then dephosphorylates it to thiamine, and then rephosphorylates it, or adds 2 phosphates at the same time, from ATP to AMP, adding a pyrophosphate all at the same time. So that's kind of interesting. I believe this is the form that we get it in our diet, but the pyrophosphate version is the kind that's actually used as a cofactor, which we actually were able to see here in this DXP synthase, using thiamine pyrophosphate as a cofactor. Yeah, I didn't get into this very much. I'm going to kind of quickly go through it. So this is what's called a Ribose Switch. So ribo Switch, and this is just a way to regulate an enzymatic pathway or protein pathway, and essentially uh, what happens is in a lot of pathways you have some kind of substrate, and then an enzyme turns it into a product, but then that product can go back and bind the enzyme and actually inhibit the enzyme, so that if you start getting too much product, then you inhibit the enzyme and you stop this process so that the product can then decrease before you start turning more substrate into product. But what a ribose switch is we have this same sort of setup here with our enzyme and our product, but instead of the product going and inhibiting the enzyme, it's actually going further back and inhibiting the mRNA that actually codes for the enzyme. So it's actually stopping that enzyme from being translated, or it can actually happen at several levels. So it can actually stop it at the transcription level, or at the translation level, or even at the splicing level. Uh, yeah, and so this is kind of a diagram of it. You can see the thiamine pyrophosphate here. You can see where it's bound to these different bases in the mRNA. So it can have the on state when the mRNA has this secondary structure, but then when thiamine pyrophosphate binds to it, it closes it up, and it causes this conformational change in the mRNA, which is then possibly that would prematurely stop the transcription, or it would inhibit translation. And I actually had a structure for that that I forgot to open up here. So where's that ribose switch? Maybe I didn't. Maybe I forgot to save that here. So anyway, I had the structure for it, but it's not important, I guess. Yeah, so I don't have the structure for that, but you can just kind of see in this diagram how that thiamine pyrophosphate goes in there. Uh, these these guanines and cysteines actually bind, uh, well, coordinate rather with the phosphates on the pyrophosphate, and then these adenines and guanines and so forth actually pi stack with the pyrimidine ring on the thiamine pyrophosphate, and so that actually intercalates into the sequence of the mRNA, and so that is how the thiamine pyrophosphate is held in there. And down here is just a list of some of the enzymes in this pathway um, that are that have this ribose switch inhibition going on. So we have our THIG, O, and C, and I'm pretty sure E is also inhibited in this way, because if I'm remembering correctly, it seemed like there was a paper that often shorthanded all of them by saying THICOGE. So this was their shorthand way of talking about the four that are being inhibited through this ribose switch, and that makes sense. The C, O, G, and E. So the G, the C, the O, and the E. Particularly the G and E are sort of necessary. They're sort of instrumental in the thiamine biosynthesis pathway. They're the enzymes that sort of bring a bunch of the other stuff together here. Uh, and so the THIO and the THIC is also inhibited, which would also make sense since this pathway has to steal intermediates from purine synthesis. So we don't, well, not we, because this is prokaryotic pathway, but the bacteria that use this pathway wouldn't want to keep doing this reaction here at the expense of purine metabolism. And so they would want to inhibit this step right here. But yeah, anyway, I think that this is already a long enough video, so I think I'm going to end it here. Yeah, I hope this was enlightening for you, and I will see you in another video.